Hello. First of all, I would like to congratulate Brian, Toby, and the organizers for organizing this very nice celebration. I'm Irene Margiolaki, and I have to admit this is very emotional for me, as I believe the role of Bob was very critical for my understanding of macromolecular powder diffraction, which I will focus on for today's very brief recording. So I came to know Bob's name when I started working at the University of Sussex in Brighton in UK as PhD student. And back then we used to use the software PC GSAS for refining fullerene-based materials. I have to admit, in the beginning, I used to hate the software because I used to find it very complex, but with years, it became my favorite, and it's the program my team and I still use for refining protein structures. In 2003, I went to work as a postdoc at the European Synchrotron Radiation Facility in Grenoble with Andy Fitz and Jonathan Wright. And during my first interview for getting the post, I was asked what kind of research I would like to pursue during my postdoc. And I answered that I had seen a few publications by Bob on protein structures refined from X-ray powder diffraction data, and I would like to pursue that difficult topic. Back then, nobody really was convinced that this area would really produce valuable results. But over the past decades, I think we know now that the technique is considered as a very valuable complementary technique in protein crystallography. And Bob started this whole era of macromolecular powder diffraction. So this was one of his first papers on whale methmyoglobin, where he showed that protein powders can really give diffraction signal, as you can see here. And it was the first case that he showed that a protein structure, in this case it's shown here, could be refined using stereochemical restraints that he introduced in GSAS, and he made available for anybody to use in the whole scientific community. The second paper I read back then refers to a new polymer of human insulin with zinc, with this molecular conformation. It's an hexamer, really. And he produced it, as far as I know, by accident, by grinding some single crystals. A new polymorph was produced, and it was the first time that Bob showed that a protein structure cannot only be refined, but really solved from powders and the technique of molecular replacement. And the third cornerstone paper by Bob is this one, where he co-crystallized lysozyme. Lysozyme is a model protein with different oligosaccharides, and he showed that from electron density maps produced from powder diffraction data, he could really identify the ligand binding sites on the surface of the protein. So these three papers started this whole scientific domain, and I always thank him for first for starting this topic, and second for sharing to the whole community for free his knowledge, tools, and the know-how in this interdisciplinary scientific domain. This was the first manual that Jonathan Wright constructed, and he made it available via the CCP14 platform, where he was trying to help beginners in protein crystallography and GSAS by providing some first guidelines on how to refine a protein structures using stereochemical restraints in both software, the GSAS software. I still give this out to my students when they start refining protein structures in GSAS. The first real synergistic result with Bob came out in 2004 as a publication. It was the structure of 30 egg white lysozyme that you see here from synchrotron X-ray powder diffraction data. We collected data at different temperatures to see the evolution of the diffraction profile, and it made it into the cover of BCA News of the British Crystallographic Association. It was also awarded in 2003 by the Biological Structures Group of the BCA. And for us, it was very important because all of a sudden people started thinking that the technique could have some impact in structural biology. Over the years at the SRF and always in communication with Bob, we tried using area detectors and analyzer crystal instruments, and we achieved gradually to improve the data quality. The profile that you see here comes from a very, very large protein polymer. The protein is called urate oxidase. It was provided to us by Sanofi Aventis in Montpellier. It forms this tetramer under specific crystallization conditions, and it gave this beautiful data. Since then, both Bob's team and our team keep producing data that really manifest that powder diffraction can provide valuable information about proteins, even as large as this monster here, from nano to microcrystals. In 2010, I moved to Patras, and we keep collaborating with Bob regardless. It's an ongoing happy collaboration with him. We had the pleasure to organize a workshop here in Patras in Greece. Here you see Bob and Camille, of course. The workshop really was the beginning for training young researchers, Greek researchers, and not only on powder and electron crystallography. And since then, I believe the number of scientists who use the technique for structural biology problems increases. It expands, of course, because more and more people get trained. Coming closer to an end, I would like to share with you some unbelievable results that we thought they were impossible to achieve back in 2000 or early 2000, but these days we know that they are possible. This is a case of virus proteins which were crystallized directly in cells, and then the cells were put in a powder diffraction, in fact, in a SAX station, small angle X-ray scattering station. Powder diffraction data were collected, and the structures of the in-cell grown crystals were identified via indexing of these profiles. This also attracted attention because it shows the many benefits, the many scientific disciplines that the technique can provide useful information. Of course, pharmaceutical proteins, for many, many years, we work with insulin, microcrystalline drugs, This is one of our very last, very recent collaborative work with Bob. We solved and refined the structure of cubic human insulin at 2.7 angstroms resolution. The model is available from the protein data bank with this code. And again, this structure solved and many more, which you can find in the literature, really suggest a mechanistic hypothesis on how insulin polymerizes according to its physicochemical, biochemical environment. So again, this is, I believe, the highest resolution protein structures we've managed to solve and refine using synchrotron powder diffraction data. Another case in continuation of one of Bob's early publications, we really pursued the potential of the technique to identify how organic ligands bind on protein surfaces, and this is very, very important for the development of pharmaceuticals. This is a more recent case where we also saw that the organic ligand and cresol can be identified from electron density maps produced from powder data and how the ligand binds on human insulin. This is a new monoclinic form of insulin, again verifying that ligand identification is also possible from powder diffraction. The synergy with Bob will never end. I sincerely hope that we'll have many, many years ahead for collaborating forces, combining our forces and our brains in order to continue this macromolecular powder diffraction era, which now we think it's ideal to be combined with serial crystallography applied at various synchrotrons. And we are very happy to see that the number of protein structures available in the protein data bank produced by us, but not only, gradually increases, which means that more and more people succeed in extracting protein structures from X-ray powder diffractions, laboratory and synchrotron data. So concluding, I would like to thank wholeheartedly Bob Dreele for all these years of his innovative research and his contribution to the whole scientific world, always sharing tools, computation tools, and knowledge with everybody with no hesitation. Bob, I hope we'll have many years ahead to continue our collaboration. Thanks for everything. You're very, very important for me, as you know, and thank you very much for listening to this very brief presentation.